Good morning, good afternoon, and good evening. Welcome back to Chapter 8. We're talking about how cells reproduce. In the previous one, we talked about binary fission, how bacterial cells reproduce. Remember that bacterial cells have a single chromosome, and therefore it's not that difficult to split it in half. The problem with splitting a eukaryotic cell in half is that they don't just have one chromosome, they have many chromosomes, and it's more difficult to make sure that you equally divide multiple chromosomes between two cells versus just dividing one chromosome between two cells. So eukaryotes have a much more complicated process for cell division. This much more complicated process is called mitosis. I hope that in high school you studied mitosis, you looked at mitosis, and you have a basic understanding of how this process takes place. The whole purpose, the whole goal behind mitosis is not necessarily to divide the cell. Mitosis is part of this process called cell division. But cell division is not 100% mitosis. There's more parts of it that aren't mitosis. Mitosis's goal specifically is to divide the DNA in half, to make sure that those two complete sets that the cell begins mitosis gets divided into two separate sets, one for each of the daughter cells. Mitosis is most concerned with dividing the DNA. If we're going to divide the DNA, if we're going to divide these chromosomes, we need to understand what a chromosome is. And when you think of a chromosome, you think of this, characteristic X shape, right? And remember that you've got a human has 46 chromosomes arranged into 23 pairs. And so here is a pair of chromosomes. And the question is, where did I get my chromosomes from? I got half of my chromosomes from my mother, and I got my other half of my chromosomes from my father, okay? So if I have 23 pairs of chromosomes, here's one of my pairs of chromosomes. How many copies of this chromosome do I have? And the obvious answer is, well, I've got 2 copies of that chromosome, right? I've got one of mom's and one of dad's. The problem is that this X shape that you're used to thinking of when you think of a chromosome is only present during mitosis. And we said a few minutes ago, we said that before the cell begins this process of mitosis, it has already doubled its DNA. It's already made a copy of its DNA. So this chromosome structure that you're used to seeing, that you're thinking about in the shape of an X, is not actually one chromosome. It's actually 2. It is one copy of mom's chromosome and a second duplicated copy of mom's chromosome. That X shape that you're seeing is actually 2 separate chromosomes attached to each other at the center. And this little belt buckle thing is called the centromere. So if each of these, if this is 1, 2 chromosomes, I don't have 2 chromosomes right here. I actually have 4. I've got 2 copies of mom's and 2 copies of dad's. These are the chromosomes that are present during mitosis. And so this is what a chromosome is. It's not actually, this chromosome is not actually just 1 chromosome. It's actually 2 copies of the same chromosome that are joined together at this belt buckle thing called the centromere. Here we see a picture. This is a single chromosome right here before we begin mitosis. Partway through the life of the cell, it's going to take this 1 chromosome and duplicate it. And now I have 2. This is the chromosome right here. This is the chromosome that begins mitosis. The process, part of the process of mitosis is splitting this thing in half so that one can go into one daughter cell and the other can go into another daughter cell. This is just one of that pair of chromosomes. Remember, there would be a second one over here. This is mom's chromosome, 2 copies of mom's. I would also have 2 copies of dad's chromosome, right? 1, 2. So we said before we begin this mitosis process that that complete set of DNA has to be duplicated. Here's 1 chromosome. I've now turned it into 2. We have 23 pairs. We have 46 chromosomes arranged into 23 pairs. All of those 46 have to be duplicated. So I don't have 46 chromosomes that begins the process of mitosis. I have 92. so that I can separate and put 46 into this daughter cell and 46 into that daughter cell. The cell that begins mitosis does not have one complete set of DNA. It has two. Humans, like most animals, are diploid. Di means two. Ploid refers to the number of chromosomes that you have. Ploidy, how many chromosomes that you have? 2 chromosomes, diploid. Some animals are tetraploid, which means they don't have 2 copies of each chromosome. They have 4. I know of a species of frog that are tetraploid. Plants aren't nearly as delicate as animals are. Animals are really delicate. We've got these really delicate things called brains that have to be made just so. We've got these delicate organs like a heart, like a liver, that have to be made just so. And if they're made a little bit wrong, you're just dead. So animals are really sensitive to having exactly the right DNA, exactly the right amount of DNA. Plants aren't nearly as sensitive about having how much DNA they have. So plants can be tetraploid, octoploid, even more than that. Plants are really flexible when it comes to how many copies of chromosomes that any given plant will have. One other thing before we go on. This process of having more than 2, this process of having more than 2 copies of each chromosome is called polyploidy. Poly means many, ploidy refers to the number of chromosomes. Polyploid means many copies of each chromosome. Polyploidy. So when you talk about tetraploid, octoploid, that's called being a polyploid. Polyploidy. Humans, we've got 46 chromosomes arranged into 23 pairs. We have 22 pairs of what we call somatic chromosomes. Soma means body, and so we have 22 body chromosomes, or rather 22 chromosomes that are dedicated to just making your body work. We have a 23rd set. That last set is called the sex chromosomes. And those sex chromosomes, we know what those are. Those are the X and the Y. And which combination of sex chromosomes you get makes the difference as to whether you stand up or sit down to pee. If you get 2 of the same sex chromosomes, 2 X's, you're a girl. If you get an X and a Y, 2 different chromosomes, you're a male. All right? 2 of the same chromosomes, you're female. 2 different chromosomes, you are male. We'll talk about sex determination in next chapter. It's not all 100% sorted out the way we just discussed. We'll talk about that later. Gorillas have 48 chromosomes. Pea plants have 14. So it is not that you are the species you are because you have a certain number of chromosomes. But it's not uncommon for different species to have different numbers of chromosomes. Though, again, the number of chromosomes you have does not make the difference as to what species you are. It's which genes you have that make that difference. The life of a cell is referred to as the cell cycle. The cell cycle is broken up into 4 sections. Let's go ahead and jump to a picture of the cell cycle, and let's look at these 4 sections. The beginning of a cell's life is right here at what we call G1. A lot of times G1 is referred to as gap phase 1, but I really prefer to have the G stand for growth. So G1 stands for growth phase 1. And this cell has just gotten done dividing. It just finished this process called mitosis. It is a brand new cell, and now it has to recover from dividing, recover from being born. All right, that's not an easy process regardless of the species. So this cell has to recover from division, and then it's going to grow. If it just divided in half, that means it's half the size it used to be. So it needs to get bigger again. So growth phase 1. It's going to grow, it's going to recover from mitosis, and it's going to do whatever job it's supposed to do. Each different cell in your body has a different job it's supposed to do. Just like each different worker in a grocery store, they have a different job. That job needs to get done. G1 is when this brand new cell begins to do its job for the first time. About halfway through the life of my brand new cell, it's going to go through what we call S phase. S stands for synthesis. Synthesis means to make. What is it going to make in S phase? It's going to make that copy of the DNA. The cell does not make a copy of its DNA right before it divides. It actually makes it not long after it was born. Roughly about halfway through its life, it's going to replicate that DNA. So it's going to go through most of its life having not one complete set of DNA, but two. Mom didn't wait until she was almost dead to photocopy her entire library of recipe books. She did that when she was 20, just before she was ready to start her family. She did it a long time ago so that she wouldn't have to do it at the end. After the S phase, we go into growth phase 2. Growth phase 2 is where now this cell is getting ready to divide. It's growing to get larger so it can divide. It's going to make copies of its organelles before it divides. It's also still going to be doing whatever job it was born to do. If it's an eye cell, it's going to detect light. If it's a stomach cell, it's going to digest food. If it's a liver cell, it's going to detoxify poisons. It's still doing that job all throughout this process. At the end of growth phase 2, we enter what's called M phase. And M stands for mitosis. And again, we said that a little while ago. M phase is not 100%. Cell division is not 100% mitosis. It's most of mitosis, but it's not all of it. The last little step here is called cytokinesis, and we'll talk